Okay, the thing is that this talk will be discussing about mitochondrion and NADH or NADPH, and is related to the energy control inside of aerobic cells. The cartoon which is here, it means that the cells, in doing their energy, or cells, must maintain a very, very delicate balance on a rather thin vertex, okay? We have to be staying here, otherwise we are in a problem. And if you want to understand that better, you need to be able to study at the same time what comes from the mitochondrion, which is reactive oxygen species and related, and also NADPH. So why mitochondrion is involved is simply when we eat, this is the lower part of the talk, you have the glucose, for example, or carbohydrates or lipids are entering the metabolism cycle to generate CO2. All the carbon ends into CO2, and the hydrogen ends into water. And that generates electrons because basically this is an oxidation reaction, and these electrons are carried to the inner wall of the mitochondrion. Mitochondrion is a bacteria, has a bacterian origin, so it has two membranes, and these electrons are shuttled, and each time one electron is shuttled through the proteins that are you find that everywhere in the books, so I will not enter in. To compensate the charge, one electron is associated to one proton that it will be transferred. Yeah, okay. That will be transferred inside of the, in between the two membranes of the mitochondria. And the concentration in protons increases. However, when you make too much electrons, for example, just after eating or and so on, you have a lot of food to process, and you feel hot because basically you make too much electrons, and this is that makes too much protons, and that will create a problem into the inner space of the mitochondrion. And it is decided to use electrons to reduce oxygen, which can go through the membrane, okay? And that makes superoxide. And superoxide, normally with the protons, will undergo into water. But about a few percent, people discuss is it 3 or 5% of the superoxide is not transformed in water, but goes into reactive oxygen species and reactive nitrogen species. Why we need this gradient of protons is because this is the way we can make the ATP. ATP, when the protons pass through the big enzyme, which is on the right part of the slide, which is ATP synthase, that allows to catalytically lock one adenosine phosphate, diphosphate, to a phosphate, and makes ATP, which after will be used in many reactions to regenerate ADP. So the problem is that if we have too much of this oxidative stress, we create ultimately something that has been studied after the Second World War, essentially, but also, for example, all the Curie family died of cancer and so on, because when you have very energetic rays, you make you split the water molecule and you generate OH dot, and this OH dot is wants to reform H2O. Why? Because the bound energy of hydrogen and oxygen in the water molecule is the strongest one in the things that are not solid. So in fact, the OH dot will be able to grab hydrogen. This morning, we spoke about peroxidation of lipids. Okay, you had some question about that. And this is exactly what is happening. But all the part, and that will generate a lot of problems that are drawn here on the slide. I will not go on each one, okay? But you will have impairment of the cell, possibly mutations, and in the best case, okay, I don't know what he's asking me to allow something. Yeah. Okay, and in the best case, the cell dies, otherwise it mutates, and that can be the origin of cancers. So, in fact, this is one of the things. Now it's blocked. Now it does not move. Ah, see, yes, come back. Sorry, can you come back? Okay, I move one now. And in fact, fortunately, we have systems in our cells or in our body between the necessary formation of superoxide and nitric oxide that create, for example, peroxynitrite. This is one in gray here near the bomb, because it's a real bomb in the or superoxide.
Recording in progress.
Okay, maybe I should stop and not give my talk. Okay? So I continue because I have good. Okay, so or metabolism. Yeah, you do that, but make that the thing works. Okay, first. Because this is not because of my voice that the thing does not work. So you have these metabolic pathways that generate the superoxide and the nitric oxide, and that makes a whole family of the primary reactive oxygen species and reactive nitrogen species. And in fact, fortunately, we have a lot of natural antioxidants. We have enzymes like a superoxide dismutase, catalase, that will remove that, so we can establish a balance, which is called the homeostatic balance, when the production, which is essential and normal, okay, I can discuss in the question why we need to have oxidative stress, and what compensate. And we have heard this morning talks about you can use supplement to help this system. So, for example, if you have here, this is in the case work we did with my friend in China about neurons. You know that 6-hydroxydopamine is the same as dopamine, essentially, except he has an OH on the ortho position or para position, okay? And that makes that when it is oxidized by oxygen, you get this quinone that now will generate hydrogen peroxide. And this hydrogen peroxide, if you have iron 2 or 3 nearby or copper, will generate OH dot, and that will create a problem. So here you have a symbolism of what is happening on the second part of the slide. You have either the 6-hydroxydopamine can enter directly through channels like those of dopamine inside of the neuron, or generate reactive oxygen species like is evident on the top, and those will proceed through the membrane. But anyway, the mitochondria inside of the neuron will receive that and become either decide that the neuron is completely non-functional. We start to generate caspase cycle and so on in order to generate apoptosis, or the mitochondria will become impaired and the neurons will be non-functional, okay? So, but now if you use an antioxidant, this is the one which is in blue here, it's called arpagide. My colleague, Chinese colleague, decided to use that because it's a natural product in China. And you see, if we measure the behavior of neurons, so we have made some micro wells, which are on the cartoon on the left, where we can identify synapse between the body of the neurons, which are in the upper compartment, and muscular cells, which are on the bottom. They create a synapse, and we have developed a very small carbon fiber nanoelectrode to slide in the synapse, and with that we can measure when the synapse is working. So on the control, you see this is a normal behavior of a synapse. The neuron is stimulated and send dopamine, adrenaline inside of the cleft, and this is detected by the electrode. Each of these bars is one burst of adrenaline that has been released. If now you put 6-hydroxydopamine, you see that the neuron become sick, cannot release. When they release very, very small amount of dopamine. You see the scale is 5 picoamp on the right. Also, it was 20 picoamp on the left. But now if you put arpagide, which is an antioxidant, this will eliminate the problem created by the 6-hydroxydopamine. And as you see, the neuron restores its own behavior. So this shows that you can bring an antioxidant from outside, which will help the problem and restore the problem. But mitochondrion is not alone, because you have this family of NADH or NADPH that are involved everywhere. This is drawing of a cell that I took from the literature, okay, because it's not complicated to draw by itself. And in the mitochondria inside of the cell. And you see everywhere you have channels that are connected through NADH and NADPH. So the problem is to know when a cell is dying from oxidative stress, you have a rise of NADH, and you have at the same time the reactive oxygen species. And who is the problem of chicken and egg? Okay, who is the first? So this is this way. So now we use the same electrodes as before, but we make them in order that we are able to detect what is needed. So on the first series of cartoon, this is the electrodes that will detect the reactive oxygen species. So we need to have nanoparticles of platinum that are deposited on the silicon carbide wire. And when I will not enter, but I can discuss later, that is sealed into the tip of Pasteur pipette, okay, extended Pasteur pipette, and then you have you insulate most of the tip, except after you dissolve with some dissolving the tip. So now the tip, which is in dark blue, is sensitive to reactive oxygen species. And you can see that here, for example, if we use antimycin A to block the electron transfer chain. That will generate reactive oxygen species. So if we use a cell with cultured with incubated with AMA, but electrode is simply silicon carbide. This is green in bottom. You have no signal. If now you have the total electrode and with the functional nanoparticles of platinum, but you let the electrode out of the cell, you will have no signal, okay? And now when you are inside, you are inserted inside of the cell, and you get the red signal. You see that you have a release, a huge release of reactive oxygen species. Now, how would you do for detecting NADH or NADPH? We use a sensor which is done with PDOT. PDOT is a polymer and electroconducting polymer, okay? So I will not enter into the whole detail, but basically it's the same. You have this electrode, and you can see we can measure that. So now we have two electrodes that are like this, and we can settle the two electrodes. How you go back? One. No. Yes, yes. So in fact, we take a pipette, which is now a double barrel pipette. So it's two pipettes glued together. Okay. In one of the barrel, you put the electrode for the reactive oxygen species. In the other, the one for the PDOT for detecting NADH. And now you have something that you see in the scanning electrochemical microscopy, something with two antennas, okay? The dimension, you have them, okay? You see they are 300 nanometer, okay? 350 nanometer, and you can penetrate inside of the cell with these two here. So I will not go into the full detail of the presentation here, but we have made all the tests. And one of the things which is important and there is a real seal of the membrane. We can detect that with also with classical system to show that the membrane of the cells is not altered after. The metabolism of the cell is not altered after. So now we can do the experiment. So we stimulate by the cells by sending resveratrol, okay? And so this is a breast cancer cell, okay? Minor, okay? And you see that the two electrodes report the rise of, for the platinum one, the rise of the reactive oxygen species. For the NADH1, the rise of NADH. And this is, here you have, if you make the integral of this system, you see that you have an integral here of the two. And you can make statistics over many cells. and you can show really that the fact that you put the resveratrol generates oxidative stress and generate also the release of NADH outside of the cell. So now, how do you solve the problem? When you look like this, the thing is I'm sorry. I have to do that because I did not plan that there will be no pointer. So, to finish, I need to thank the people. So, now I have two positions, one in Paris, which is because I am emeritus in Paris, and I have a position of invited professor in Xiamen University. This is on the side, but most of the work was done with Wuhan University, with the excellent team of Professor Weiwa Wang, and most of this work was done by the student Yuting Jiao, who is really, really excellent. We have a paper now in Nature Nanotech done with her, which should be released. So I will stop here and thank you for your attention. And in the bottom, except for Xiamen, and on the vertical side, you have all the academies in which I am a member, the International and French Academy. Thank you all for your attention.